All right. Okay. So switching gears now. So moving forward from the cytoskeleton, we're going to go ahead and jump in on our lecture about molecular motors. So specifically today, the learning objectives are to talk about the molecular motives kinesin and dynein. So these are the microtubule specific molecular motors, and we'll talk about the actin associated molecular motor next lecture, myosin. We'll talk a little bit about why cargo transport is important compared to just general diffusion in the cell, a little bit about the mechanics of these, and our disease application today is rabies viral pathology. So I know we haven't talked about specific families of viruses all too much yet. So this is a little bit of a diagram showcasing the viral family tree. So we have different DNA or RNA viruses. Obviously, everybody's familiar with SARS-CoV-2. That's a coronavirus. Here, let me get my laser pointer. Coronavirus, so that's one of these RNA viruses. And the rabies virus is part of the rhabdoviridæ family. Sorry, can't speak today. So this is an RNA virus, a single strand, and it has a helical type DNA. This virus also is a cylindrical virus with an envelope protein. And on the outside of this envelope protein are different glycoprotein spikes. And so similar to SARS-CoV-2, this is what it's using to enter cells. And specifically, this is a neurotropic virus. So this means that it binds preferentially to neurons or neural cells. So in terms of what these glycoproteins are targeting, we'll talk about this more later in the lecture, but these are neuron-specific extracellular proteins. So I'm sure rabies is familiar to everyone in this room. It is most commonly transmitted through animal to human transmission, and so it can be considered a zoonotic infection. So usually this is if an animal that's carrying rabies bites you, but it can also be transmitted through aerosolized particles, similar to SARS-CoV-2, with somebody that has the infection. And so the symptoms of this make it a little bit of a tricky infection. So similar to other viral infections, I'm sure we're really familiar, there's generally an incubation phase once a virus enters your body. And this incubation phase is so that the viruses can enter certain cells and start replicating, multiplying themselves, creating more. And usually this process is without any symptoms. So for rabies infection, this incubation phase can be from days to months, even up to a year. So that makes it really tricky if you did get bit by an animal to not know whether or not you actually got rabies as a result of that. But following the incubation phase, we have something called the prodrome phase. And this is when the virus infects your central nervous system. So a couple of the symptoms that are caused from this are fever, fatigue, itching at that bite site if it was recent, and different GI abnormalities. And this process usually happens anywhere from 2 to 10 days. So now it's becoming a little more obvious that there has been a rabies infection. And then following this prodrome phase, we have the neurologic phase. And so this is when the virus actually makes it up to your brain, starts infecting different neural cells in your brain, such as in your hippocampus, your brainstem, other nerve cells. And so this can cause kind of the canonical rabies symptom, which is hydrophobia or fear of water. But this also causes a lot of other more serious symptoms, such as seizures, hallucination, disorientation, paralysis. And this phase is relatively short as well before ultimately leading to a coma or fatality. So this is a really deadly disease, in particular because once the infection has caught on, there's nothing to stop or reverse it. So there's not a waiting out the infection, it's not going to end well, and there's nothing to reverse it once it's already started. But thankfully, in the US, a lot of animals are vaccinated for rabies. So specifically, dogs are one of the main transmitters, but dogs that people own are generally vaccinated for rabies. So this is more of a danger in countries where there's wild dogs or other types of animals that are not being vaccinated. So, like I said, the only treatment for rabies is immediately post-exposure prophylaxis. So there is a vaccine for rabies, and so if there is treatment with this vaccine immediately after getting bit, before the viral infection can really take hold, then it is treatable. And this is actually something that Louis Pasteur developed. So this was part of what was attributed to his rise to fame, his being known as the father of microbiology, is that he developed the first rabies vaccine and delivered it to a young child immediately following a bite from a rabid dog. All right, so we'll get a little bit back into the overlap between rabies and molecular motors at the end of the lecture. But we're gonna go ahead and jump in, talk about why do we need molecular motors? Why can't things just diffuse naturally around the cell and find their appropriate location? So that would be the only other option if there wasn't any kind of directed transport. And this diffusion happens as kind of a random walk. So this figure on the left here is showing a theoretical path of random motion in any kind of 3D space, or 2D space in this case. So if the x-axis is some distance, the y-axis is some distance, you start at the middle, this particle might move randomly, and as you can see, there's not really any direction to it. And this is driven by Brownian motion. So this is thermal energy driven motion. And everything in the cell has some Brownian motion. So there's always fluctuations in particles and proteins in the cell membrane within the cell. And so this theoretically could drive these particles to move where they need to go. So if we were to quantify how far these particles can go, this figure on the right here is kind of a graph of this. So it's plotting the average distance or absolute displacement. So in a positive direction, how far away are these particles moving from where they start? And so you can see it'll go up and down, get closer and further away. But if you plot kind of a fit line to it, it looks a little bit like what the square root function would look like. So this means that it can get to places that are relatively close in a reasonable time. But if you try to go a very long distance, it'll take a very, very long time. So we can model this in terms of random diffusion, movement of particles due to Brownian motion. And so this equation on the top right here is how we would model this. You're not responsible for knowing, for regurgitating this equation, but it's good to know some of the components that are integrated in this and what are the driving forces of this. So this equation is known as the Stokes-Einstein equation. So these two guys, Stokes and Einstein, developed it. Einstein is the famous Albert Einstein, and this was actually his graduate thesis work. So everybody hopes their graduate thesis work will be as memorable and have a big contribution like theirs, but we can't all be Einstein, I suppose. So this equation on the left here, the x squared with these triangular brackets, it stands for the mean square distance. So x is distance. This is being squared so that it's positive, and then just taking the mean over time. And the different constants here, or components of this equation, are listed. So the Boltzmann constant is always a constant. This is kind of derived from statistical mechanics. And this is given as unit of energy per temperature. So this is the inherent amount of energy at a given temperature from this Brownian motion. The T is absolute temperature, so this is in Kelvin, so there's no negatives here. There's no absolute zero in absolute temperatures. It's always going to be above zero. And then here we have the eta term. This is the viscosity of the fluid. So this is playing a big role in terms of the transport within that fluid. And then the R is the radius of the particle. So this can be anything. This could be, we're thinking about a theoretical protein or some ion or something within the cell, for example, or just any kind of particle. What size is that particle? And then the N here is dimensionality. So how many directions are we moving this particle in? Is it going along a line? Is it going in a 2D plane? Is it in a 3D space where we have all directions? And this equation can be transformed to be in the form of time. So in this case, the equation is now solving for how much time would it take to travel some distance. So we can use this equation to calculate in a cell how long would it take for some particle to move across the distance of a cell. So one thing to consider, especially for viscosity here, I think this might be the least intuitive in this equation, that the viscosity of cytoplasm with everything in it is about 1,000 times that of water. So if you're just imagining a particle moving in water, which we would most likely think of, it's going to be way different than particles moving within the cytoplasm. So if we do this calculation, we take a ribosome, which is a fairly large protein, and calculate how long it would take to diffuse the length of an axon. And some of these motor neurons are about a full meter in length. So this is a little bit on the extreme side, but in this example, this is something that would genuinely happen in your body, and it would take 700,000 years for this ribosome to diffuse this length. So this is just a little exercise to hopefully appreciate the importance of this cargo transport and the role of these molecular motors in the cell. Okay, so on to the content about molecular motors. What are they? From the pre-lecture, you might remember these are enzymes, and they're enzymes with the primary function of converting chemical energy into work. So this work is a force times a distance. And so the energy that they're often using can be many different forms. And this is often ATP, the canonical energy source of the cell, but GTP is often substituted for this, as we saw with cytoskeleton polymerization. Electrochemical gradients can also be utilized. And so there's one really important molecular motor that we'll talk about later in the lecture that's actually the enzyme that creates ATP that utilizes electrochemical gradients, because can't use ATP to make ATP. So this is a really important function of our cells. But for many of these molecular motors, the specific work that is being done is motion. And so this can be either transport of cargo or movement or contractility of cells. And so today we'll talk a lot about transport of cargo, since this is one of the primary roles of microtubule-based motors in the cell, although you'll see microtubule-based motors can also contribute to motion, as well as actin-based molecular motors can still contribute to transport of cargo, but we'll be talking about them in these different categories for the purpose of the lectures. All right, so the general mechanism for how these motors work is that they are taking in, binding some energy source. ATP is often this energy source, like I said. And then the enzymatic activity of these molecular motors will cleave one of the phosphate groups from ATP to ADP, and this drives a conformational change in this protein that then allows another binding step onto the filament and can move this process forward. And so you can see the example here for either myosin or kinesin that are working in different ways, but are both binding ATP, changing their conformation, rebinding, and moving forward in this way. So another way we can think about these molecular motors in terms of their mechanism or how we would define them is with this concept of processivity. So processive motors are ones that are moving continuously without releasing from the cytoskeleton or the filament that they're attached to. So you can see with these two examples that kinesin is always attached. So when one part of the motor unbinds, the other is still attached, and it will rebind before unattaching the other one. Whereas the myosin in this example is detaching and then reattaching again with the binding of a new ATP molecule. So this isn't necessarily true for all myosins. There's a lot of different types of myosin motors, and we'll talk about those in the next lecture. But this is just an important concept to be aware of when we talk about defining how these molecular motors work. So some more concepts from the pre-lecture. There's a lot of other aspects of these molecular motors that we want to define and understand. So in particular, how fast they move, or the velocity, just the distance over time, is an important factor in terms of how fast they're moving cargo. How large of a step does it take? This might influence how much energy it takes to get from one point of the cell to another. And we can define this in terms of the step size. So this is how we would talk about how large these steps are. How much force they generate. So how much force is required to do this protein conformation kind of depends, especially with cell motility and migration. If you think about that movie we watched in the last lecture, the cell pushing things out of the way, the force can be really important for this. And so we can talk about this in a couple different ways. With isometric force, this is just how much force can be applied to a stationary object. And this comes up a lot in terms of exercise. Isometric exercises is just pushing on something that's still. But average force is more applicable for a wide variety of things. So this is the average force that is exerted on these filaments over time with each of these steps. So there's lots of other types of ways we quantify these motors. So the duty ratio is just how long are they on, and this is important for a lot of different types of machines. So these molecular motors are pretty much just protein machines. So we can define these in the same way. And how quickly is it utilizing the ATP? So this is the enzymatic activity or the speed of this enzyme reaction in terms of what is propelling this movement. So these are ATPases, so this would be the ATPase rate or the cycle time, since each time ATP is hydrolyzed, it goes through one of these cycles or steps along the cytoskeleton filament. So these words can be interchangeable in this case. Okay, so these different types of molecular motors are going to associate with different cytoskeleton tracks. And like I mentioned, we're going to be diving in a little bit more into the microtubule-based motors today, so the kinesins and the dyneins. Next lecture, we'll talk about actin-myosin interactions. But there's generally not motors that are running on intermediate filaments. So does anyone want to volunteer why that is? Yeah. Exactly. So they don't have a polarity to them. So there's no way to know which direction you're moving, and this makes them not very useful for transport inside the cell. All right. So these microtubules and actin filaments, in terms of just the polymerization of these filaments, they can also sometimes be considered molecular motors themselves. And this is because of the Brownian motion within a cell. So we talked a little bit about this in terms of the actin polymerization at the edge of a cell, pushing a cell forward. And so this can work in kind of a thermal ratchet mechanism. So this animation just shows kind of how this is done. So the red balls here are maybe one monomer of actin, and this black line is the cell cytoskeleton. So because of this Brownian motion, everything's moving in the cell, even the cell cytoplasm. And so there's going to be some polymerization happening of actin that goes all the way up until where the cell cytoplasm is. But because of this movement, if the cell cytoplasm is moved away from this actin polymer, when there's an addition of a new monomer, then it'll push that cell, the cell wall forward, and it'll help propel the cell forward in motion. Yes. Yeah. Yeah, so the word thermal ratchet is kind of just referring to the fact that it's driven by this thermal motion, the Brownian motion, and ratchet is just meaning like with each new step, there's a movement. And so in this case, the step is the addition of a new actin monomer. And so we can imagine this at the edge of a cell where actin has polymerized and it has reached the end of the cell cytoplasm, it's hit that lipid bilayer. But even though it hits the lipid bilayer, that bilayer is not staying still. It's moving back and forth. So there's some opportunity in that time where a new actin monomer can come in and associate with the growing end of this actin chain, and that will further push the cell, the lipid bilayer forward, and then this process can repeat over and over again to kind of push that lipid bilayer and allow the cell to move in that direction.
One thing I was gonna say, one analogy I've always kind of liked about the thermal ratchet model is you can almost envision this as like a really rowdy concert where like people in the front row are like sort of pushing the front railing and like more people slide in and push it further. I'm sure y'all will go around. You can imagine a really rowdy concert. It's kind of the same thing, like people are sliding in, able to push it further, and like the more monomers of people come in, it's easier to push even further in.
Great. So coming back now to these microtubule-based motors specifically, we have these two general types, the kinesins and the dyneins. And so the difference in them primarily, or one way you can think about their difference, is their directionality, or which way they move in the cell. So the kinesins are going to be plus end directed. So you can see these here, they're towards the negative end, moving towards the plus, and the dyneins are the opposite. They're moving towards the minus end. Both of these have a motor domain and a cargo binding domain, and we can refer to this as the head and the tail. So the motor domain is this part that's attached to the microtubule, and it looks kind of like a head in the schematic. It's like a little oval, where the tail is what is binding the cargo in both of these. And the machinery for this is different, so we'll talk about that in more detail, but just general terminology to get the sides of these motors straight, you can refer to them as heads and tails. And there's two different kinds of dyneins. We'll talk about this more in later slides. There's cytoplasmic dyneins that are doing this cargo transport, and there's axonemal dyneins that are involved in ciliary and flagella motion, and we'll talk about how some of this works briefly. So the reason why we need these two different types of microtubule-directed motors is so that we can get all along these microtubules, because they're organizing themselves around these microtubule organizing centers. They can either go outwards from this or in towards the microtubule organizing center, depending on what molecular motor you're using. So this can be important in the protein trafficking cascade or in endocytosis if you're going the other way. So if you're producing a protein in the cell starting at the rough ER, you're going to need to go down towards the microtubule organizing center in this example, and then back out to the cell, and that will require two different types of molecular motors. And the same thing with the endocytosis in this case. So if there's something that was taken in from out of the cell and needs to go up towards the center of the cell, then you would need to utilize a dynein molecular motor in that case. Okay, so a little bit about the structure of these molecular motors. There's the cargo binding tail that we mentioned. And so that's this part. In this example, it's the N-terminus of this protein. And then there's this multi-domain AAA ring, and this helps facilitate some of the movement of this protein. So it allows it to bend and change conformation. And this part of the protein is actually where the ATP is binding and hydrolyzing, and the conformation is changing. And so this makes sense that it would be in the middle, connecting the cargo binding tail and the microtubule binding head. But there's also this stalk region in between that just transmits that force that comes from the ATPase activity. So this tail, as you can see up here, is generally going to associate with a number of adapter proteins that are ultimately going to be what is binding that cargo, and we'll talk about why that is in later slides. So kinesin is really similar. It's again got the molecular or the microtubule binding motor head domain. So this schematic's flipped in this case. We have the microtubule at the top. So this part here is now the head, and this part here is the tail. And so this has this head region that binds the microtubule. It has this neck and hinge, and it has this coil-coil conformation. So it's a little bit different in terms of the rigidness and the overall components of this motor, but they both have this cargo binding tail and microtubule binding head. So in terms of regulation, we'll talk about more types of regulation when we talk about actin-myosin, but for these cargo binding molecular motors, there's one main regulatory mechanism that determines whether or not they turn on or off. And this is actually the binding of the cargo. So when they're in their inactive state, they're kind of folded up like this, and that makes the microtubule binding part hidden and shielded on the inside of the protein. But then once the cargo binds, it changes its conformation, and then it opens up. And so now this top portion here is able to bind the microtubule. So this is for kinesin specifically, but there's a similar cargo-mediated activation mechanism for dynein. Yes. Exactly. Yeah, cargo is anything that's being transported. And we'll talk about some of those different cargos more specifically. And so for kinesins in particular, the cargo binding domain, because it is a part of the protein, is inherent to the primary structure or the amino acid sequence of that protein. So for that reason, kinesins are part of a superfamily that can bind a lot of different types of cargo, where each type of kinesin is specific for that type of cargo. So this schematic here is showing these different domains lined up with the similarities and differences. So you can see in this case, the green, the motor domain, is relatively conserved across these different types of kinesins. And even this coiled domain is always present, but the length of it is different, whereas this cargo binding domain can look really different. There can be a lot of different types of structures here, and this is because it's going to be carrying a wide variety of cargoes. So this figure here shows what this looks like in terms of their actual tertiary structure, what it looks like when they're fully formed. And these are some electromicrograph images and some projected schematics of what they might look like. So you can see the shapes of these will vary widely, but the main components of them will stay the same. Yes. So it gives the flexibility and the ability for the molecular motor to move. Good question. Yeah, that's the main purpose of the coils. So this slide just kind of highlights all of some of the range of these cargoes that can be carried. You don't have to remember what these are or where they're present in the cell, but this is just to hopefully give you some more information about these and help you appreciate what the superfamily of kinesins can bind to and transport in the cell. So dyneins, on the other hand, are not part of a superfamily. There's basically just one that is doing this cargo transport. There's one, the dynein 1 is the primary dynein that's utilized for cargo transport. Dynein 2 is specific to cilia and flagellar cells, where the axonemal dyneins are the ones that are creating the motion in the cilia and flagella. But this dynein 1 is doing a lot of the transport in different types of cells for different types of cargo. And it can do this because, unlike kinesin, where the cargo binding domain is inherent to the protein, dynein utilizes different adapter proteins. So it's going to associate with different proteins that can bind the appropriate cargo. And so one important part of this is the role of dynactin. And so this binds to dynein and can help facilitate the binding of some of these other adapter proteins. So this will help with binding the cargo ultimately. Yes. Or is it like one? Yeah, good question. So dynein 1 and dynein 2 are different proteins. So they're not the different parts of the same protein. They're different amino acid structures that are going to be transcribed in different types of cells. So this axonemal dynein, we talked a little bit about how this cargo transport works in terms of the binding of ATP and the movement along these microtubules. And the axonemal dyneins work similarly, but they're not binding cargo. They're facilitating movement. And so how this looks briefly is there'll be these pairs of microtubules. So like you can see in the schematic, there are two microtubule chains in green that are lined up together. And then you have the dynein motor end attached to one side and what would otherwise be the cargo binding end attached to another microtubule. And as they can move these microtubules and they can offset each other. And so this causes a bending. And then the other one can move and it can do that in similar ways. So this would cause, for these structures, there's many of these duplicate pairs of tubulin tracks with these dyneins in between them. And so the bending of these can facilitate the overall movement when it's coordinated. And so what's really interesting is in flagella, because you have different types of bending all along this structure, there's different activating and inhibiting signals that can run throughout this entire structure that can cause these microtubules to bend the structure or stop bending the structure. And this allows the coordinated movement of these multi-unit structures to facilitate this movement. So you can see in this active form, these microtubules are bound at both sides, and so this will be causing this bending, whereas in this inhibited side, it's unbound here on the bottom. And so that gives it some freedom to bend in the opposite direction and to be driven by this active side that's creating the bending. All right, any questions about this before we move on? Okay, switching back to the other type of dyneins, that was a little aside in terms of how the dyneins are creating movement, but the cargo transport is the main thing that we're talking about. So cargo transport in dyneins is, again, facilitated by these adapter proteins. And these adapter proteins can bind a wide variety of things. So again, similar to kinesins, this is a schematic that just shows how many of these different types of cargoes can be facilitated by dynein transport. And something to appreciate here is that these are not just small proteins. So entire mitochondria and ribosomes can be transported. And again, like we talked about, this would not be possible with just the use of diffusion. So the transport and binding of dyneins really makes a lot of these actions happen. And transitioning now back to rabies, this is important in a lot of viral-based diseases because there's many types of viruses that can hijack this dynein transportation through these different adapter proteins in order to leverage the cytoskeleton network and move throughout cells. All right, so like I mentioned, this is not necessarily specific to one type of virus. This is just a big table that lists a lot of different types of viruses that can utilize this mechanism. And so rabies is listed here, and this is for the purpose of transporting in the cell. So a little bit about how this happens, getting to the mechanism, the pathogenesis of rabies infection. So you encountered some rabid animal that bites you, and this is usually going to be occurring in your muscle of some sort. So the rabies virus, RABV is how it's shorthanded, will be infected within that muscle, and when it's in the muscle is where that incubation period happens. So this is where the virus is replicating, it's creating more of itself, transcribing its genome, creating all the different types of proteins it needs to form more of itself, and then it can endocytose from these cells by fusing with the plasma membrane of the muscle cells that it's in. And so it'll do this in places where there are specific neuromuscular junctions. So these are the places where our motor neurons are connected to muscles, and this is what is sending the signal from your brain to your muscle to fire that muscle to move it. And because these viruses have a high affinity for neural cells, when it finds one of these neuromuscular junctions and it endocytoses into this space here called the synaptic cleft, it can recognize a neuron through the binding of both an NCAM molecule, so this is that cell adhesion molecule, and this receptor known as P75NTR. So the NCAM helps facilitate its recognition to this spot, but the P75NTR will facilitate the endocytosis of this virus into this neuron, this motor neuron specifically. Once it's endocytosed into this P75NTR, it's a transmembrane protein. So there's a part of this protein that is accessible on the inside of the cell, and this part of the protein that is accessible on the inside of the cell will bind to a certain dynein adapter protein. And this allows for the transport of this cargo all the way up this motor neuron. So this motor neuron will connect to the central nervous system. It can do the same mechanism, moving from the from this motor neuron to the central nervous system, all the way up the spinal cord, and then eventually to the brain. So these NCAM molecules, if you remember, they're specific to neurons, the neural cell adhesion molecule. And so this can facilitate the movement across many different types of neurons and in long distances across your body. All right, any questions about this? Yeah. Yeah, so that is both what's causing the endocytosis and is binding it to the dynein for the transport up the axon. All right, so if there wasn't any more questions, if there's more, we can go back to it. But this binding of different viruses to our molecular motors, specifically these neuron-specific molecular motors, can be a really useful tool that we can leverage in the lab. So this is something known as neuronal tracing or synaptic tracing. And this can allow you to trace neuron paths within the brain. And so this would be by having some virus that utilizes different types of molecular motors and following it throughout the cell. So you can attach some fluorescent molecule or some kind of tracker that allows you to visualize the location of this virus particle. And you can see where it goes in the brain or in these neural circuits. So there's different types of tracers or terminology that we can use for this. There's an anterograde or a retrograde. So this is an example of a retrograde, where retrograde just refers to movement towards the nucleus. And it's retro because generally how these neurons are working are they're going from the dendrites, which are the little protrusions near the nucleus, down the axon. So this retrograde movement is going the opposite direction. So for the specific types of dyes where these viruses are utilizing this retrograde motion, you can see if you inject some dye with these viruses in one spot, you can maybe ask the question, what neurons are connecting to this region of the brain, and track backwards as a result of that. You can also do the opposite. You could do a retrograde tracer. So this, an anterograde tracer. So this is utilizing the normal direction of signaling that these neurons will do. So if there's a cluster of neuronal cell bodies and you want to know what portions of the brain are this cluster of neurons connected to, you can inject these viruses that are going to hijack this cargo transport in that portion of the brain connected to their fluorescent dye. And then you can see where it's going to move to from there. What portions of the brain are these dyes going to transport to? So this can get really fancy. You can utilize some types of viruses that are going to go in both directions. If you want to see connections in every direction, you can do something called a transsynaptic tracing reagent. So these are reagents that will jump over synapse. So they'll go from one neuron to the next neuron it's connected to the next neuron after that, next neuron after that. You can see an entire circuit of neuron signaling, or in this example, this would be a non-transsynaptic tracing region. So this would just go from the end of one cell to the other end of that cell. And so this can be really useful in terms of figuring out these brain circuits. So how are different neurons connected to each other? How does this influence thought or these different regions of our brain? what's going on in terms of the neurons that are influenced in these roles. And so this can be a really useful tool. It's utilized a lot in mice, but it can hopefully get us a little bit closer to understanding the brain as a whole. So this link here is called Neuron Browser. It's from the Howard Hughes Medical Institute. And if this is something you're interested in, you can peruse this a little bit further. So just to end here, we'll finish a little bit early. There's a bunch of faculty at UVA that are researching these kinds of things. So Anna Cliff is looking at viral latency and reactivation, axon and dendrite trafficking, or this idea of neuronal tracing. All right, so next week, yeah, we'll have a couple announcements.
So I've had 3 quick announcements before I lose your attention or lose you and go to your next class or next minute. So for the quiz question on the violence factor, I didn't realize I edited the slides and actually took out that phrase. So I'm going to give that question back to you guys. That's on me. I'm sorry about that. I'm sure that was probably a painful moment. Saying the phrase you didn't actually hear much. I went back and tried to listen to the first intro portion, and so when I said it was in the intro part, it used to be, and it wasn't in what I had here. So that's on me. The next thing is we have an assignment due next week on Tuesday. So so you guys identified your biological problem. The teaching team is going to finish giving you guys feedback by tomorrow. The assignment that's due on Tuesday is just you taking the biological disease and problem, and then just providing a hypothesis, maybe a few sentences of background that sets up the hypothesis. What I would highly recommend, again, we want to stick with primary sources here. We want to stay away from Wikipedia. Wikipedia can be a really helpful tool if there's something about the disease, like if you come across the phrase virulence factor, you don't know what that is. You can go and look that up on Wikipedia, that's fine. But to understand your biological problem or disease, I would highly recommend using PubMed or Google Scholar to find primary sources. Review papers are great to get a sense of what's the current landscape of what's known and what's not known. And that dynamic of what's known and what's not known, it's going to be super helpful for you to navigate how you identify what your proposed mechanism will be or your proposed alternative therapy. So it'll be really helpful for you to identify to describe what the disease is, what's known, what's not known, maybe what the current gold standard of treatment is, why that doesn't work or why that's not great, and what you think is going to be a better idea that could improve things. And so I will post that assignment with those details later this afternoon. The last piece is we have an exam next week. So Thursday we have the exam. We're going to first start with a quiz. And again, if you are happy with your, we're going to keep posting the quiz grades to keep you guys updated on how you're doing. If you're happy with your quiz grades and if you're happy with how the quiz goes on Thursday, you will be dismissed and excused after the quiz and after we go over it, so you get a sense of how you did. And then following that, maybe 5-ish minutes after the quiz is done, we'll go ahead and transition and start the exam. The exam is just going to be, we're going to have what, 4 or 5 lectures in this unit? Mm-hmm. So we'll have a 30 to 35 question exam. That's going to be all very similar questions to the quizzes. And so what's also going to happen is that the teaching team is going to coordinate for there to be a review session to take place next week. But again, I'm also going to make a plug here. I bet you guys know what I'm going to say. Office hours are super helpful for this. The office hours that I've been doing have been in my office 9 to 11 Wednesdays. I'm going to try and switch that to be in a conference room, because that's typically what I did in the past few years, where usually, again, make a plug here, 8 to 10 people would always come, and we'd have an opportunity to go through things. And so there'll be a review session. There's going to be all of our office hours to be available to you guys to help out. So just to recap, quiz question goes back to you. Assignment is due Tuesday, and I'll have that posted up tonight. And then exam next Thursday with a review session during the week. Yes. Yes, we can do that. 24 hours beforehand. Yeah. Okay, we'll shoot for 24 hours beforehand. Are there any other questions? Yes. Yeah, so this is a great question. Hold on. So someone asked a really great question. I don't mean to stop. We still have time. What if your hypothesis lines up with current research? So That's gonna happen. So there's gonna be an idea you might have that you submit for Tuesday. You might find out in about, let's see, we're in February. You might find out in 1 month that as you read more papers, that this idea has been done. This also happens in real science and real research. So what's gonna end up happening is we're just gonna help you. We're gonna help you and recommend that based on what you know of the literature about your area, that then maybe you can pivot and try and identify like, okay, if this has been done, how can I propose something that's that hasn't been done or that's novel? That is not like a huge shift from what you were already proposed and all the work you put into it. But like, how can we shift it so that it maybe builds on what's already known to make it a new novel idea approach? Does that help make sense? And again, the idea is that these assignments are to help keep you guys on track. There's going to be a point where you realize, I don't like this idea. And again, that happens in science. That happens during my PhD. Maybe happens or has happened during your PhD. So that's okay if you think my idea sucks or it's already been done. That both of those feelings happen in real science. And what our job is going to be as a teaching team is to help you guys pivot or maybe come up with something that's a little more compelling or that builds on the existing work to be something that's new and novel. All right. Again, like there's an opportunity here to try and come up with something new and novel and exciting based on a subject area that you guys are interested in and that you guys identified. So it's going to be our job to help you guys. But it's also going to be incumbent upon you guys to try and to put in the legwork, to try and read the literature, read review articles, read primary research articles, and to get a sense of what's already done, what's already known, what's not known. That was a really great question. I appreciate you asking. Are there any other great questions out there? I also love dumb questions because they're really easy to answer, but I also don't want you to feel like it's a dumb question. I feel like I throw myself in the foot with that comment. All right, with that, thanks for your time.